So my name is Nick Holway. I work for a company called Novartis, or a pharmaceutical company, and I work in the early stages of drug discovery and research. So the actual bit I work in is this one up here, which is Novartis Institutes for Biomedical Research and NIBR. I'm pointing this out because we use the terms NIBR and research interchangeably, just to confuse people. So I've been working at the company for 10 years, first in the UK and here in Switzerland now. So I'm going to start off with a big statement, and hopefully through the course of this talk, I'm going to justify the statement about how important high-performance computing is to us. So HPC means that we can get better, more targeted drugs to patients quicker. So this is basically what my group and the other equivalent groups within Novartis do. So what I'm talking about today is, first of all, I'm going to introduce the company very quickly. I'm guessing most people in the room will not be very familiar with drug discovery, so I have a few words on that. What our high-performance computing looks like, and then I'm going to share some examples of our scientists' work that they've done on our clusters. And then finally, I'm going to speak about what we think is going to happen soon in our industry. So very quickly, we are a large $50 billion or so drug company. We have various units, eye care, generics, and pharmaceuticals, which is the bit I'm working in. And also cancer has been split off, even though it's more or less the same thing as pharma. So we've got about 6,000 scientists in our company, 500 clinical trials, 90 drug candidates, and about 400 research projects ongoing at any one time. So the interesting bit to most people in the room is that we have about 400 or so computational scientists working in research. This number is growing all the time. And also we have an increasing number of scientists who are using computation on the cluster but don't actually realize it. So we also operate in, create drugs for a number of different areas. So things like if you have transplants or heart disease, cancer, asthma. So we cover most areas of drugs. So it's very... I'm not a corporate person, that was the corporate introduction over. So this is a bit about drug discovery. So pharmaceutical drug is a drug used to diagnose, cure, treat, or disease. So some examples of drugs and how they work. So if you look at cancer, as for an example, some cancer drugs will disrupt the cell's ability to divide and also to actually move around as well, which will obviously stop the cancer growing. In theory, the downside to this is that these side effects are not very good. Also, you can disrupt a bacterial cell's walls to act as an antibiotic, and also, or in depression, you can alter the way chemicals are taken up into the brain. So this is what we do here. So what we're trying to do is to get a drug, we say onto the market, which basically means patients are taking the drugs in the clinic. Where I work is over here in discovery. This is a very, very long process, so sort of 10 to 15 years, a bit longer. So whatever we do here, if we can speed this section up, it means that patients can benefit from our drugs over here. And as, of course, we are a for-profit company, the longer we have here, before the patent expires, which would be about here on this diagram, obviously the more money we make, and that keeps our shareholders happy. So as you can see, there's a very large rate of attrition. So I'd say it's a bit more than, I've nicked this slide from someone else, but I'd say that number is actually bigger. So we have lots of compounds here for a potential target of drug, and over time they get knocked out quicker. So one thing that we want to do is to fail quicker, which sounds odd. If we can fail here, then we're saving a lot of time, effort, and money. And if we can say stop a drug here because of bad side effects, then we need never ever trouble a patient with the very first clinical trials. So later on, I will talk slightly about the HPC here, but it's not what I'm going to be talking about today at all. So what does our high-performance computing look like? It's very similar to those of you who run a university with a biology or chemistry department. The only real difference is that our nodes tend to have a lot of RAM. This is because we have a lot of bioinformaticians running. We also have some specialized nodes with GPUs and also with a lot of RAM, which is mostly for genome assembly. Storage, our data is mostly generated in the Windows land, but the vast majority of our computational work, they work in Linux land, so we need dual protocol exports. So that's where our data lives, but when we are actually doing the computation, we tend to use Lustre for that, which speeds things up very nicely. So in terms of software, Red Hat or CentOS, Grid Engine, we use Bright for deployment. Our software is built with easy builds, we use modules, various compilers, various normal libraries available as well. In terms of languages, C++, Fortran, CUDA. So I've made Python and R bold because most of our scientists will program in Python or R to run on the cluster. There's not actually that much new high-performance code as many people in the room would see it being written. We do run other people's code, which is glued together by this. We also have some people who like using MATLAB because it's very nice to program in. So as I've said, yes, the software stack is identical on our scientists' workstations and also what we call scientific servers. This is a server which is dedicated towards being used by our scientists as opposed to, say, a database server or a DNS server. So our concept is that you can take some code that you've written on your Linux desktop in one of our sites, and then you can then, assuming you've done nothing stupid, of course, you can then run this code anywhere on the planet. So that could be Shanghai, it could be Basel, which is where I work, or one of our American sites. That, believe it or not, took a long time to fix, 3 years, to actually get all the software harmonized globally. It was the most boring, least glamorous project, but it was absolutely essential for us to work. So this is our team. So we are the Scientific Computing Group. I would probably call us SciComp quite a lot. As I've mentioned, we are represented throughout the planet, and we are a combination of systems administrators, mathematicians, and scientists like myself. So a couple of years ago, we won an HPC Wire Award for our work of cloud computing. So elsewhere in research, other teams look after storage and servers and workstations and so on. So one thing that our scientists, including the SciComp, have done in the last few years is try to build a community up. So we've worked very hard at this. It started off with a load of scientists thinking we should get together and speak over lunch, and then over time it ballooned. And then this year we've actually managed to get formal funding out of the company. So we have fortnightly seminars, social events, beer in the pub. We had a deep learning boot camp recently. It was very similar to the one yesterday afternoon. And recently we also had a 24-hour global workshop starting off in Shanghai, going through to California via video conference. It was kind of cool. So interestingly, this came from the grassroots, not from the top down, which is probably why it's been taken up properly. Amusingly, we had a seminar day once, and we didn't have any funding to the last minute, so we had a baking committee and a scientific committee. In the end, we got funding, but the baking committee stayed, so we all baked cakes for the seminar day, which was really nice, actually. So, as I've mentioned, I work in research. However, elsewhere in our company, we have other people working in high-performance computing in a separate but similar environment. They do things like modeling the drugs in the body. So the first time that one of our drug hits a human, some people will have modeled it in a high-performance computing infrastructure. They process data in clinical trials, and they do some very interesting work around where drugs go in the body with fluid dynamics. But unfortunately, I'm not here to talk about that today. So these systems are actually much more tightly controlled than our own, with lots of validation, testing, as you'd want if your drug dose is being decided. So, I'm going to talk now about a couple of examples from our scientists on how we use high-performance computing in drug discovery. So, there's lots of ways people use molecular dynamics, the bioinformaticians do their NGS, ligand protein docking. We've been having that running for years and years and years. Image analysis has become important in the last few years, and I'm going to be focusing on that in a moment. And in the last year or two, cryo-EM is becoming very interesting. As I said earlier, we're very similar to biology or chemistry department. But the examples I'm going to use today are testing drugs in a very special kind of assay, which is quite new to us. So traditionally, our scientists in the earlier stages of research, they basically do chemistry, but in a very, very small format. So those of you might remember from your secondary school looking at doing an experiment in chemistry where you would say, see how much compound changes color, for example, with change of pH, that kind of thing. So our scientists traditionally would use that kind of assay, but miniaturized in these things called plates, which is a phrase I use a lot. They're plastic. They're about the size of my hand with lots of different holes in, which are called wells. So that's how it used to work in the early stages of drug research. However, what's happened, that obviously gives you a very reductionist approach. You need to know the particular thing that you're looking at. So what in recent years they've started to do in a very automated manner is to grow cells or small organisms like malaria parasite and then see how they react to drugs or stimuli or anything else. So we call this phenotypic assay based on the cell's phenotype. And this means the scientists can see what the drug is doing for an entire cell or cells or population of cells, which gives you quite a lot of more information about things like toxicity, how a cell might work, or if you're looking at a subset of cells. So this is an example I pinched from the internet, from the cell profiler group. So you can see here, these are cells on both sides here. This is called a scratch assay because someone will have got a pipette tip and scratched down here. And then 24 hours later, you can see how the cells have moved across. So this is a very easy example of how you could use this kind of assay to promote wound healing. Of course, if you're looking at a cancer assay, then these cells moving is a bad thing, so you might want to see less of this. It just depends entirely on what the scientists are trying to show. So high-content screening is a variant of this approach, where with lots of miniaturization and robotics. It's very, very interesting to go and have a look at, actually, if you get the chance. So they'll look at various cells, things like malaria parasites, and this generates quite a lot of data. So they can use videos, and they can also take millions of images in a single experiment. So the biggest of our instruments, we estimate, generates about 600 terabytes a year, which is raw data and also derived data, i.e. intermediate things, output. So one example of this, which one of the scientists has generously let me use, is many of you will remember the ice bucket challenge that went very popular on social media a while ago for ALS, or motor neuron disease, if you're on this side of the Atlantic. So our scientists have been looking into this for a few years, and one of the things they're trying to do was to create an experiment to see how well drugs work. And for this, they found the best thing to do was use GPU. So in this disease, what we call junctions between the cells that excite the muscle cells, they basically don't work properly. So we'd like to create a drug which corrects this. So to do this, they put some motor neurons, some nerves basically, and what we call myotubes, muscle fibers in English. They were grown together, so I'm translating this in real time out of biology into English, in a plate, and to this they add drugs to see which works. So they're actually imaged in real time to see how quickly these cells are contracting. This is actually very, very hard to see, as I'll show you in a second. And also it's actually quite hard to segment using a computer. So what we have here are these cells that I was on about earlier. So what I want you to do is to ignore the one here that's going to flash and take all your attention in a second. And just look at this figure on the left-hand side. So you can see that there's occasional sudden movements, say about here. I'll show you that again. You don't need to be a scientific genius to realize that measuring this manually is incredibly tedious and error-prone. So what Imtiaz did when he was working in our group, along with a lot of other scientists, is they started to segment this using an optic flow approach, which is what we have on the right-hand figure here. So you can see that this is instantly easier to visualize, and of course we now have it's now quantitated as well. So again, this is I'll show you this. So this is a different field here, but you can see roughly here we have two areas which are contracting. What you have to look at here is, this is actually quite a hard figure to read. So the color indicates the direction the cells are moving in. And down here you can see when we are picking up the cellular contractivity. And here we have all the various regions on the cells. So this was all done on a GPU. So he used, excuse me. Okay. So this by using a GPU to do the optic flow calculation, he managed to get an increase of about 80 times over using a normal CPU. This figure was derived a few years ago, so with the newer Intel GPUs, it might actually be, this 80 times might actually be too low an estimate these days. But as with most of our assays, they are embarrassingly parallel. So an individual slower GPU isn't too important. What they want are lots of them, and they get their parallelism through the scheduler. So it was a very good project. We had bench scientists who now know what high-performance computing is and what GPUs are. We have lab automation experts because these things are miniaturized. And also we have some informaticians, both in the scientific computing group and also in the business itself, all working together to create this. And the end result is our scientists have a new method of investigating the early stages of drugs for motor neuron disease. So moving on to another one. This was actually published, and I know some people in the room have mentioned this from the same scientists. So that, again, we're talking about high content screening, HCS, and they use deep learning for this. So as I mentioned earlier, high content screening is normally used it's normally performed using tools such as Cell Profiler from the Broad Institute, or Fiji, which is basically ImageJ, or a number of commercial tools which usually come with the instruments. However, these require a lot of fiddling, they need to be reasonably good at image processing to be able to use these tools. And also, some of the time, it's not possible to actually go through using these tools to actually work out what the cells are doing. So we got a project working here using deep learning to look at these kinds of cells. So they were looking at CNNs to derive the image's phenotypes. And what they're able to do was to use the image's pixel intensities with no previous knowledge on public and our own private data sets. What they're able to show is that using these data sets and deep learning is they were classified better than the normal methods. And they were actually able to show a quantitative response to some of our drugs. So basically you have drug response as an S-shaped sigmoidal curve. Which is the kind of thing that our scientists are actually looking for. Also, there's no need to design a unique pipeline for the image processing, which saves some scientist time, which is actually quite valuable to us. So I've obviously gone over this at a very, very high level, but if you're interested, then this work is actually published in the journal Bioinformatics, and there's some code and also the original paper. And I'm not expecting you to remember that at all, but if you search for William Godinez et al., then you should be able to find it. So my last thing that I'm going to be discussing for our HPC examples is how we push high-performance computing to normal scientists. So most of scientists, they're not trained in computers. If they're a biologist, they've probably done almost no maths at all, and they definitely can't program. Chemists are a bit more mathematical, but only just, but most of them cannot program. But what is happening is their data is getting too big to crunch on a single workstation. So some of them will buy what they call their supercomputer, which everyone in the room would call their workstation under their desk with, these days, maybe 30 cores or something like that, and try again. It eventually gets to the point that they just have too much data to crunch, but what they don't have is the expertise to crunch it. So although I said at the start we have 400 scientists, informatics scientists, they are a small proportion of our scientists, and they're very, very busy. So what we've tried to do is to give the scientists the tools that they can use high-performance computing, but with no expertise required. So again, I'm using high-current screening as an example, and we use Cell Profiler a lot from the Broad Institute, which is a GUI tool where they can just point, click, and create a pipeline for segmenting their images. And from this, they can then upload it to a server at a central site, and then we have a tool which they use to visualize their images, and within that tool now they have a button that they can click, and they select a pipeline, and then they go. So this is quite similar in some ways to the iBrain 2 project, which some of the people in Switzerland will know about. So this is a few screenshots, which is not projecting very well at all. So anyway, you can have to use your imagination that they are they're selecting their images here, they go through, choose some options, and then here this is basically saying what is running or not running. You can see it's green. Green means it's run successfully, red there's a problem. They can go through, and this, if they can get their data back a lot quicker. So prior to this kind of tool, they used a server provided by the software vendor. It was quite a big server for the time with about 40 cores, and it would take 3 months to get their data back. By using high-performance computing, they're getting their data back within a few days. So another approach for the same problem is this chap here, Jonas, has spent a lot of time abusing the tool Jenkins for continuous integration of software. So what he has is a system where it's very similar to the one before, where you can upload a cell profiler, but he's also got R running off it. And again, you're launching jobs on the cluster from a nice web-based GUI. So in this case, this work has been published. You're probably quicker if you search for his name than remembering this. So we've made the code open source here, if anyone wants to play with it or contributes to it. So it's there to play with. Jonas has done a lot of talks on the internet if you search in the videos as well, if you're interested in this approach. So that's this is the final part of my talk. This is basically where I think we're going to go. So deep learning is absolutely exploding at the moment. There are lots of interest in lots of different fields. So currently we're running on GPUs, but in time we may find that other platforms also work very well for us. We are agnostic, we will run whatever. So cryo-EM, which is a new technique for structural biology for deriving structures from images from electron microscopes, is also looking like it's becoming very big. We are putting in quite a few GPUs and also extra scratch into Basel to accommodate a new facility soon. We're also looking into collecting and processing data from clinical trials in real time or near real time. So traditionally, the clinical trials, you'll get the data back. It'll be something like blood pressure or how far a patient can walk in 6 minutes, and these are all very slow tests. So we're seeing devices coming up. So I've injured my leg and I'm wearing a belt that we normally get to clinical trials, but this is not in real time. I have 2 months worth of data on a memory card that gets uploaded, runs on the HPC cluster, absolutely fine, no problems whatsoever. But if we start having, say, data streaming from a mobile phone or something similar to it, Internet of Things, then we're going to need to be able to collect this in real time. I imagine that whatever systems we use won't look like the HPC we have, but most of the concepts will be very similar. So we're also looking into using some of the so-called big data technologies, such as Apache Spark, into high-performance computing, and DK will be talking about this kind of thing in a moment. Also, we are looking into containers. We've looked at various containers. It's looking a lot like we're going to use Singularity, but we haven't made a final decision yet. We're also running high-performance computing in the cloud for certain kinds of workloads, but mostly for when the cluster is too busy, or when we have embarrassingly parallel jobs without a huge amount of data. Things like ligand protein docking works exceptionally well because it's a few gigabytes of data and a lot of computation, and just the more calls, virtual calls you throw at it, the better. So thank you very much for your attention. Has anyone got any questions?